Hello, my name is Marcus Shearer, and today I'll be presenting my final biology laboratory report, researching and comparing the microbiomes of bean beetles. These experiments were conducted at Morehouse College in the year of 2024, and the first images I'd like to present on the left-hand side is our microscopic imaging of our first incubated plate, and on the right-hand side is our plate results from when we began our initial green pea sample incubation. Let's get started. I'd like to start with a student background about myself. Again, my name is Marcus Shearer. I'm a first year biology major at Morehouse College. I'm from Dallas, Texas. I also have an interest in health science as well as an interest in the medical field. I'm also very enthusiastic towards lab related experimentation as well as research oriented experimentation. An introduction. The microbiome, which refers to the total genetic material of microorganisms within a defined environment, has become a focal point in scientific research. Investigations into diverse microbiomes extend to various species, a popular one including marine animals. Studies on marine animals have recognized them as a sentinel species for their capacity to adapt to oceanic environments similar to terrestrial mammals. Examination of their skin and organs has unveiled similar microbiomes, suggesting a correlation between microbial communities and animal well being. Likewise, research on coral microbiomes underscores their symbiotic bond with corals and their role in acquiring nutrients without disrupting the ecosystem. While microbiome studies using coral or marine animals are much more initial and popular studies, they shed guidance on experimentation of microbiomes, thus paving the way to studies such as the one I present to you today, which involve bean beetles. Short background of bean beetles. Originating from Africa and Asia, bean beetles have a short lifespan, primarily dedicated to feeding and reproduction, with females laying eggs and dried beans, leading to larval development inside the seeds. As larvae mature, they consume the bean's contents before pupating and emerging as adults. Male beetles then seek female mates, initiating a reproductive cycle that sustains their population within a 10 to 14-day lifespan. Intent of research. In our lab, our research focuses on bean beetles and their reproductive processes and microbiomes in mung beans and green peas, which me and my partner have chosen. We use data from past experiments to explore how environmental factors impact the hatching and DNA traits of bean beetles. By examining the DNA of bean beetles from various bean types, such as mung beans and green peas, we aim to reveal insights into potential genetic adaptations and evolutionary shifts in these agricultural pests. This work not only enhances our knowledge of insect biology, but also opens avenues for novel strategies in pest control as well as crop safeguarding. Hypothesis. We are investigating the question, how do the microbiomes of sibling and offspring beetles compare? Our hypothesis, crafted by my partner and I, suggests that variations in microbes will arise from the green pea and mung bean beetles having distinct hosts as well as distinct microbiomes. Some materials include centrifuge tubes, micropipettes, electrophoresis system, a microcentrifuge tube, several agar plates, incubators, sterile pestles, refrigerators, a DNA cyber subway system, as well as the R-Shiny system, a thermal microvolume 1C nanodrop spectrophotometer, and other miscellaneous lab materials. How do our materials aid us in experimentation? Well, the centrifuge tubes are used for spinning samples at high speeds to separate components based on density, such as isolating DNA or proteins. Our micropipettes are utilized for precise measurements and transfer small volumes of liquids, crucial for preparing samples and reagents in molecular biology experiments. Our electrophoresis system is used for separating and analyzing DNA fragments based on their size and charge, essential for studying genetic variations or identifying specific genes. Our microcentrifuge tubes, similar to centrifuge tubes, but are designed for smaller sample volumes and are commonly used for various molecular biology applications like DNA extraction as well as purification. Several agar plates are used for culturing and growing microorganisms, including bacteria for fungi, to study the growth patterns, interactions, and susceptibility to treatments. Incubators are used for maintaining specific temperature and humidity conditions to promote the growth of microorganisms or cells in cultures. Sterile pestles, used for grinding and homogenizing samples, such as breaking down tissues or cells in DNA extraction or protein analysis. Or refrigerators, used for storing reagents, samples, and culture plates at controlled temperatures to preserve their integrity and stability. Continued materials. Our DNA subway is used to analyze bioinformatics. Our thermomicrovolume spectrophotometer is a precise instrument used for measuring the concentration and purity of nucleic acid samples, crucial for assessing DNA or RNA quality before downstream applications. Other miscellaneous lab materials may include gloves, coats, buffers, chemicals, and safety equipment, which are all essential for conducting experiments safely and effectively. Each material plays a critical role in different stages of the experiment, from sample preparation and analysis to later data interpretation and storage. This all ensures accurate and reliable results in our bean beetle research. Methods. Methods include experimental setup, such as obtaining beetle populations, beetle division, depending of selected project, variable manipulation, such as food availability for the beetles. Moving on, we have data collection, PCR, gel electrophoresis, then statistical analysis, documentation, and finally, sequencing with the help of our Shiny and DNA Subway. On the right hand side shows figure 1 and 2, which compare both green pea and mung bean beetles after being crushed for our initial sample isolations. Experimentation. During the experiment, we conducted bean beetle sterilization and extracted DNA from both the green pea and mung bean beetles using a nanodrop. Surface sterilization of the bean beetles involved treating both green pea and mung bean beetles with disinfectants. We first immersed the beetles in 10% bleach for 3 seconds, followed by sterilized water for 10 seconds, 70% ethanol for 5 seconds, and sterile water for an additional 10 seconds. Subsequently, we transferred both batches of beetles into a microcentrifuge tube and allowed them to mix overnight with the formula for extraction. In the following class, we proceeded to crush the beetles using a sterile pestle in a liquid mixture containing 450 microliters of 0.9 sterile saline solution, preparing for microbiota culturing process. After centrifugation, we applied 100 microliters of microbiota extract serum onto nutrient agar plates. We then combined 40 microliters of the remaining microbiota extract with 360 microliters of 0.9 sterile saline. Continued experimentation. Following this, we plated 100 microliters of the extract onto nutrient agar, EMB plates, and PA plates, and placed 100 microliters of sterile saline onto the negative control plate before introducing additional chemicals. We then documented all the DNA by crushing the beetles and analyzing it with the nanodrop. And on the right-hand side shows our Figure 3 enlarged full concentration BA plate taken on February 14th, 2024. PCR. Once initial samples were incubated, me and my partner performed our PCR for the first green pea sample. PCR, or polymerase chain reaction, is a revolutionary method developed by Kary Mullis in the 1980s. PCR is based on using the ability of DNA polymerase to then synthesize new strands of DNA complementary to the offered template strand. We began by pipetting 13.5 microliters of PCR mix into tubes, then selecting one bacteria colony to collect a sample from, then placing it in 100 microliters of sterile water. Then, 11.5 microliters of the bacteria were suspended in a tube, becoming our genomic DNA. This DNA was heated at 95 degrees Celsius for 30-second intervals, repeated 3 times, and centrifuged until a final sample was captured in our new tubes. Data. Figure 1. First nanodrop reading shows adequate levels of sample concentration of green pea sample 1, showing about 28% concentration. Figure 2 shows our second nanodrop reading, which has an exceptional concentration from our mung bean sample 1, showing about 132 concentration. Results. Regrettably, our group was unable to finish gel electrophoresis, microculturing, or an R-shiny/DNA subway analysis due to time constraints and limited supplies. I believe that if we had completed the remaining methods, our group would have produced excellent results due to our high levels of concentration across all of our samples. Discussion. Despite encountering limitations that prevented us from conducting all required experiments for a thorough sample, the data we did collect remains highly valuable for future scientific and research endeavors. A crucial discovery from our study is the distinct variations in microbiomes between sibling and offspring beetles. This outcome supports our hypothesis that such differences would emerge because of the beetles having diverse hosts. These discrepancies in microbiomes carry significant implications for advancing our understanding of insect biology, evolutionary adjustments, and interactions between hosts and microbes. And while we encountered such limitations, my partner and I do not deem our experiments a failure, as further research could deem them very important in the overall instincts and crucial discoveries of our hypothesis. Further questioning. Would our DNA synthesis reflect our previous findings on hypothesis? How would our data have compared to other students who are conducting the same experiment? How would our findings compare to the data of different beetles? Broader impacts. Advancing in this field is imperative to uncovering the intricacies of microbial communities within insects and their influence on ecological dynamics. As we delve into the variations and interactions of microbiomes with hosts can inspire novel approaches in agriculture, disease control, and ecological preservation. In essence, our experiment sets the groundwork for upcoming inquiries into the complex relationships between insects and their microbial companions, ultimately fostering progress in entomology, microbiology, and ecological research. Final consensus. Investigating the microbiomes of bean beetles offers opportunities to delve into microbial diversity, ecological dynamics among insect communities, and the potential impact of microbiomes on pest control methods. Moreover, our discoveries add to the expanding understanding of how microbiomes influence organismal well-being and functionality. To then summarize, our study provides insights into bean beetle microbiomes, emphasizing distinctions between sibling and offspring beetles. How can such experiments impact our future? Well, studying bean beetle microbiomes can assist scientists in various future experiments by providing a comparative framework for understanding microbial diversity and interactions across species. The insights gained can then be applied to other insects and even vertebrates, aiding in the design of experiments on microbiome host interactions. Additionally, this research can inform ecosystem level studies, contribute to biotechnological applications, and offer evolutionary insights, as well as enhancing our understanding of ecosystem processes and resilience. Some acknowledgments would include myself, Marcus Shearer, as well as my lab partner, Knowledge Ederson, Professor Greer, our fellow students, and the Morehouse College Biology Department. I would also like to give a special thank you to Professor Greer for your invaluable guidance and support throughout this semester. Your expertise and mentorship have been instrumental in our successful experimentation on bean beetles. Your insightful feedback and encouragement have fueled our passion for scientific inquiry and discovery. We are grateful for the opportunity to learn from you and to contribute to the field of biology under your mentorship. Thank you once again for your dedication to our academic growth and for making this semester a very rewarding experience. Here's a list of our references used, and I would like to give a one last thank you to our audience for your attention and engagement during this presentation. We appreciate your interest in the fascinating world of bean beetle microbiomes and their implications for science.